bioinformatics at Harvard, and I'm the founding chair of that new department. And so the meta message that I'm going to have in this talk is to invite you to much more seriously consider getting yourself involved in medical applications. Because not only does it represent 1/6 of the United States economy, that gets results certainly no better than investments that are half that or a quarter of that in other parts of the world, but lots of your family members are hurting and will be hurt and will not be helped because of the brokenness. And when I was asked to speak here, I actually thought what I could do. And the first thing would be the usual thing I do, which is to essentially, in the academic fashion, surreptitiously brag about the great things we do. And so I'll do that now, very quickly as a preamble. And so this will be a coy way of saying the things that are worth bragging about, but I won't go into detail because I actually have another agenda for today. But for example, I could tell you about how we're using all the medical corpora of the world, all the full text, all the databases of claims databases to allow them to just stick in natural language text into a web browser and get a differential diagnosis that's as good as pretty much any doctor that can be used by patients, and that we're going to do an artificial but important sociological test show that it can pass part one of the medical boards, which one of my grad students thought of, because even though I thought that was a silly test, he had the good marketing idea that rather than just showing some machine learning performance, just showing that we can outperform average medical students would be a signature achievement. I can also talk to you about the way we've used electronic health record systems across the nation to divine unpack, for example, the nature of autism and subcategories that are not really recognized by clinicians, but are being more recognized by parents. I could talk to you about how we are using claims data to actually do better than genetics prediction of the likelihood of a particular child being born with a particular disease. But I'm not going to do it with that. Instead, I'm going to use the fact that we have a very smart audience here. And I'm going to put you to work. I'm going to tell you a true story after a little bit of introduction. And I'm not going to have the answers. It's going to be I hope, reasonably clear articulation of a methodological problem that I have. And I'm going to invite you to think about it. We'll discuss it together. And I have really, again, two agendas. One, the first agenda is very forthright, which is I want help. The second agenda is to, again, get you more involved and interested in these medical applications. So I call this, how can we know that we're helping precision medicine with rare cases? And let me give you a bit of context, because there's a bunch of words that look like English, but are in fact jargon. In fact, when I was doing my PhD in computer science, what was initially very confusing is that a lot of words were English, but were overloaded with terminology that computer scientists meant, and it was not obvious to me until I was much deeper into my PhD. Okay, this is not working. Do I have to get up? Nope, I just have to go and press buttons. So, precision medicine, what the hell is that? So I was on the National Academy of Science Committee that actually wrote the Precision Medicine Report. For the young people who don't know what these National Academy reports look like, they're basically books that end up as PDFs, and although you can pay for them on Amazon, you can just go to the National Academy Press, nap.edu site, and download the reports. And if you're furthermore lazy like I am, you can just read the executive report, which is always about less than 20 pages. And so what we wrote in this precision medicine report, and find the agenda, which led me actually to be at the White House with President Obama when he announced the Precision Medicine Initiative, What we said was, well, we said this a little bit indirectly, we have Google it. Google has taken this relatively boring thing that no one cared about back when I was a student, namely geography, specifically longitude and latitude. Using those common coordinates, they've layered on top of it multiple data types and created an ecosystem such that every young person now really cares about geography, because knowing where your friends are, where the traffic is, where the weather is now informing us daily. So geography is omnipresent in a way it wasn't previously. And we said, could we do something like that for medicine? And we noted the fact that we were then, in 2011, at the cusp of the breakthrough, the price breakthroughs in genomes, where you can now measure 3 billion bases in a human being for less than $1,000. And we said, okay, we're able to measure the genome and the way the genome is chemically modified in non-genetic ways, and our microbial friends, and our signs and symptoms, and our exposures. And there's a bunch of studies that individually report on that. But if we could put them all together into a common coordinate system so that we could actually have a multi-axis view of patients, we could understand much more precisely what that patient was about. And there's lots of evidence, which I will not go into today, that shows that we can actually do that well. And as a result, there's several large projects that have taken that on. And the underlying hubristic notion is that we're going to be able to retaxonomize medicine, reclassify medicine, based not on signs and symptoms. Because indeed, if you look at most of the names of our diseases, the names of the diseases are the symptoms. Diabetes mellitus, what's that? Sweet piss. Rheumatoid arthritis, inflamed joints. And therefore, since the names are actually the names of the signs, not the names of the causes, it reveals that we actually don't know what the cause of disease is, and therefore we're only treating symptomatically. Just keep you awake. What is the one disease area where we actually know the name of the cause, and therefore we name the disease after it? Infectious disease.
AIDS.
What?
AIDS.
Age? Age is not a disease.
No, not AIDS. AIDS.
AIDS? AIDS? Yeah, AIDS is an infectious disease.
Right.
So for medicine, if it has anything to crow about, is how it's conquered, at least temporarily, infectious disease. It's because we actually understood the causes of disease, and we were able to treat it etiologically. As opposed to diabetes, we don't treat the cause of diabetes. We treat blood sugar, rheumatoid arthritis. We don't treat inflammation. We don't treat the cause of inflammation. We treat inflammation. And so that's one part of this. The other part is to understand that standard medicine is not measuring all things we need. And this diagram that we published in the Journal of the American Medical Association just made the point that there are structured and unstructured data of various types which contribute to our understanding of health. And so that, for example, if I took an identical twin of mine, and so therefore I knew the genomics, and I knew some of the clinical findings, it would not be as useful as knowing whether my twin or I had a fitness club membership and were actually going to the fitness club. That would change our risk stratification enormously compared to all these types. And so we published this in a viewpoint to make the point that A, think beyond standard clinical data types, and B, that there's a bit of a linkage problem, which is another methodological discussion we will not have today, which is how do you link across these data types? There's been, in public health, a long bemoaning of the fact that we don't have a universal health identifier, which is opposed on various grounds, including by some civil rights folks. And certainly there are countries such as Denmark which have a universal health identifier, but that does not help you in linking to these other non-clinical data types. So I'm just putting it out there that linkage is an important thing. Last of all, I want to point out that, and I've shown the slides in many contexts, but for those of you who haven't seen it, it's important to bring them up to speed, just to show you how broken medicine is. So again, 3 years ago, or 4 years ago, a plane went down in the Indian Ocean, Air Malaysia. CNN was covering it 24/7 for 2 weeks, which was great because it was not Trump 24/7, but it was Air Malaysia, and it's absolutely appropriate. An Airbus full of passengers had disappeared. I mean, we didn't know why. And so that's really worthy. So what if I told you that in Boston, just out of 2 hospitals, arguably our 2 best hospitals, we had 700 plane loads of passengers, AKA patients, who disappeared and we didn't know it. And that's that. This is an 18%, 17% increase in heart attacks that happened, and we were completely unaware of it until we actually did the analysis of the electronic health record. What's worse, unlike Air Malaysia, maybe, we actually shut down our Airbuses because that's Vioxx. And if you do the case control, it was of exposure to a drug called Vioxx, a very good drug that doesn't cause much gastric disturbance. That was great for treatment of osteoarthritis pain, but the marketing folks at Merck said, let's use it now for everything, and it got used extremely widely, and we killed a bunch of people, and we didn't know it. So that tells you have a system that's not thoughtful and not self-monitoring, and that's how bad medicine is. Furthermore, it's knowable, because at the first arrow, Kaiser Permanente in North California was looking at the literature and made the judgment call that there was some risks associated, and they took it off formulary. So they saved their patients this craziness. And by the way, this happens all the time. I can tell you that this was done retrospectively, so you can, say exactly. Retrospective scope is all well good. What about the future? But I can tell you that there was a controversy about a drug called Avandia, rosiglitazone, and we were able to look into our database. It's an oral hypoglycemic due to diabetes. We were able to look into our database and do the analysis, which is actually tricky because it's observational data, but basically it came up with an odds ratio of 50% relative to a drug that had very similar chemical structure, same indication, so there's no confounding by indication. And as a result of our study, the FDA black boxed this drug. But again, if you don't look, you're not going to know, and we still don't look.
Merck also lied.
What?
Merck also lied. And
Absolutely. But my point is, even though they lied, if we'd been self-aware,
yeah,
we should have been able to see what the heck is going on with this increase in heart. Why are we seeing an increase in heart attacks? And it wouldn't have taken much to look at their medication and see, even with multiple hypothesis testing, what was the residual odds ratio that was significant after Bonferroni correction. It still would have worked.
Mm-hmm.
It's pretty pathetic. Okay, last of all, I want to give you a glimpse of the future. So 7% of the population has a rare disease. So each disease itself is rare, but we have so many of them that it can be up to 7% of the rare population, rare disease population. Something interesting happened. There's a program called the Undiagnosed Disease Program that came out of the NIH, UDP, and it was very simple. It said, we're going to get an exome. So the genome is 3 billion bases. 1.5% is the part that codes for a protein. That's 150 million bases. And so back in the day when it was still expensive, you do the exome. That's the coding part, the part that makes protein. And we would do the exome on these patients who had not been diagnosed forever. They were suffering. We didn't know what was wrong with them. And some of them were dying, and we didn't know what was going on. They were very expensive, by the way. And just by getting these patients to the right expert and looking at the genome or the exome, the NIH, within their program, were able to get a meaningful diagnosis, that is a life-changing diagnosis in about, depending how you count, 40 to 60% of patients. That was enormous. But what really was enormous was in the middle of the Obama era, with a lot of partisan strife, there was a bipartisan agreement because of the following. Patients from each constituency were saying, I want to be seen by this program, because it turned out a lot of people are suffering from these undiagnosed diseases. And there was a bipartisan agreement to blow this up to a $250 million program and create 7 academic health centers, which would be seeing these patients, and would do genome or exome sequencing for all of them. And the first thing that was, and although ultimately it funded these clinical sites and 2 sequencing sites and model organism sites where you basically using gene editing CRISPR, you create the mutations that you think you found that are clinically causative into flies and zebrafish to see if it actually causes disease. So ultimately it was all that, but the first thing that was funded was the coordinating center, and I applied to be a coordinating center. And unlike typical statistical coordinating centers, I said we're going to do something sociologically challenging. We're going to take all the identified data and we're gonna put it, including clinical identifying data, pretty sensitive stuff, and the genomic data, all identified, and put it into the cloud, Amazon cloud, and make sure it was HIPAA identified. I'm sure you've heard of HIPAA. And then something else, which I had never heard of, maybe some of you have heard of, FISMA, I had never heard of it. So if you're working with federal data, you have to be compliant with a whole new set of standards that Beltway bandits will charge you a lot of money to certify you on. And why did we have to do that? Because one of our clinical sites is the NIH. So NIH is part of the federal government, so that data was federal data. And so I said, we're gonna shove all the data into the cloud. And so we're gonna triage these patients. They're gonna come in through the Harvard website, through our coordinating center. And they get triaged to these different sites. And wherever they show up, the data will show up with us. And it'll be accessible, identified to any authorized investigator / clinician throughout. And it just took us 1 year. I know that for some of you that sounds very long. I can assure you in NIH, federal government speed, that's instantaneous. We had all the IRBs, we also had all the tech transfer offices of all these universities say, no stickiness. The patient can go anywhere and we don't own them as intellectual property. We also had to navigate some very invisible lines to me that are now very bright, which is IRB's institutional review boards do not like commingling clinical care and research. But by definition, these were all commingled, and so we had to have IRBs just accept that. In fairness, we had so much money behind us, and these institutions wanted so much to do with it, that was a huge bribe, essentially, to make them very, very willing to move forward. And that's what happened. So let me tell you, in that context, about a common diagnosis that became an undiagnosed rare disease. And this is a story about the child of one of our faculty. So the faculty is one of my long-term time friends. He's a Harvard Medical School professor. And his very bright young child, about 14, 15 years ago, at age 3 years and 10 months, started having blood in his stools. And he underwent endoscopy, put a fiber optic scope up your rear end, and they found inflammation throughout the colon. And so he was diagnosed with something called ulcerative colitis, which is a type of inflammatory bowel disease. But he did very well. He was started on a very mild anti-inflammatory agent called sulfasalazine. And because my friend reads a lot of stuff, he decided that fish oil was important as well. So he started on fish oil. And he did well for 10 years. And then when he went through puberty, as is often the case, he flared. So something about growth hormone and testosterone just does the trick. And he flares. And what does flaring mean? It means you have pain and you start pooping a lot. Initially, he was only about pooping about 6 to 8 times a day, which is embarrassing but not insurmountable. And by the way, I want to time myself well. I have until
1:30, but that includes question.
Good. That's very generous. Well, let me try to shoot for 1. And so let me invite questions. If there's anything, I recognize it's not a medical audience, and so there's something medical that's not obvious, just interrupt me. Second of all, if something is methodologically does not look kosher, which there will be a lot of non-kosher. By the way, thanks for the kosher food. And I will be very open about the non-kosher methodology that was driven of necessity, and I'll tell you about the hidden assumptions that may not be obvious to you. I'll unpack it all. But as you'll see at the end of it, I'm going to ask you for help. So he flared, and he was started on prednisone, high-dose steroids, and they helped when they were high, but when you brought them down to sustainable maintenance levels, he flared up again. Try to screw around with this microbiome, the bugs, the germs in your gut that we live with, which by the way have more DNA than you have in the rest of your body. There's more DNA in your host bacteria than in your own cells. But so we tried to screw around with a vancomycin, a harsh IV antibiotic, and that helped a bit, and then it got worse. We started him on, so there's inflammation, and inflammatory cells are white blood cells, and they proliferate. So if you treat them with something that looks like an anti-cancer agent, in fact isn't an anti-cancer agent, 6-mercaptopurine, which screws around with DNA replication, that's often a treatment, but that did not help this kid after 3 months. And he started pooping more and more. In fact, he started pooping every hour. At that point, when you poop every hour, you're no longer sleeping. So this kid who was doing very well at Brookline High School here in Boston, Brookline rather, was no longer going to school. And they just got a dog, and he's no longer playing with the dog. He's just lying in bed. And by the way, just sociologically, I should note that it's very awkward. It's already awkward if you're a patient to actually talk back to doctors. Let's face it, it's all awkward. But when you're a doctor, it's even more awkward because basically you ostensibly know what you're saying. So when you're challenging them, you're actually not just saying, I'm an idiot, I'm challenging, you're saying, I'm an expert, and I think you're wrong. And so in order to avoid that awkwardness, I was going with my friend's wife to the doctor, to the GI specialist without him. So I was being heavy. And I was going, so I could say bluntly, and so if they didn't like what I said, it was just Zach that they were annoyed with. By the way,
This is
That's
actually
mine.
your phone.
that's mine.
You're so busy.
Actually, it's very clear. I recognize it's a number that's in the same first three-digit prefix as mine. That's always telemarketing. They figure out how to get to it. Brilliant. Anyway, so yes, so I was the heavy, and he started on this what's called the first generation antibody. So it's made a huge difference. These antibodies that specifically attack a protein, a molecule. In this case, it was TNF, which is a molecule that causes a lot of inflammation. And companies who found this have a gold mine because I've been told that the worldwide market for this per year is $70 billion for these TNF inhibitors. You, through his insurance, were paying $150,000 a year for this drug. And this is a relatively common disease, inflammatory bowel disease. But it didn't help. And so the conversations with the GI doctors were going to, okay, let's go to something that's quote curative. Let's remove the colon. Now that's a big deal because it's removing the colon. Not only that, you have a 14-year-old and it's a 3-part operation. You have to create a colostomy first before you, because to let the other part heal before you bring it back in and re-hook it up. So it's a nasty thing. I mean, if you told me as a teenager that I'm gonna go around smelling like a little bit of poop around here, I might shoot myself. But in any case, so I told my friend to hold off. And by the way, it's not actually curative. It's curative for some, but there's still a little bit of colon tissue right near the anus left, and that can be inflamed, and you still might need meds, so it's not perfect. I told him to hold off. And so we pushed harder in ways that were quite exploratory and that were not standard. Furthermore, let me tell you, he and I were on long phone calls in ways that patients never do because, except with a few exceptions, they don't know they can do it. But you can't actually can. With people at MGH, Brigham, UCSD, and what was amazing is everybody had a different way of treating this. There was no standard of care. There was really no standard of care. And that's going to be important as it relates to the applied statistics question I'm going to give for you, not as a puzzle, but as a
That
genuine.
might be a good thing if you have no idea what's going on, because maybe one will work.
Well, I think it reflects the fact that nothing works well. And in fact, I didn't mean to say this at this point, but I'll say it, that turns out probably about half patients ultimately fail to respond to these medications. But that's not the way it's presented in the marketing materials. It's not even the way it's presented to us in medical school. And so he was trying on a fancy antibiotic that is for which we know no resistant organisms, so you have to get special papal dispensation to use it, this drug rifaximin. And then we went on to a second generation monoclonal antibody, and on a pretty heavy dose of a immunosuppressant, which gave him a little relief, but not much. And this is not an immunosuppressant you want to be on for a long time because it's been associated with a lot of infections and so on. And so this brings us to, so this second monoclonal was added. And this brings us to December. So at this point, the mother has given in. She accepts, I mean, she can't see her kids suffering anymore. And she accepts that they're going to have surgery in January. I'm begging my friend not to do it because it seems like a big step from which there's no return. And he's caught between me and his wife, which is not the best place to be. And then he appeals to me to do something. And he said, I saw the following presentation of yours. So this is something I had done a year and a half before. So, basic biology. We have the same genome in every single cell in our body, more or less. That's changing every day now, but more, in fact, one of my faculty showed that as your brain develops, they did it by single cell sequencing throughout the brain. As the brain develops, little mutations are inserted, mistakes, as you go, and so you can actually retrace the phylogeny of your development of your brain just by sequencing through the genome of your brain. But more or less, the 3 billion bases, are the same. But what's not the same in each cell in your body, especially a liver cell from a skin cell from a brain cell, is which part of that program is running. So if you want to use a computer metaphor, what's in the registers and in RAM is the actual running program. The source code is, or the compiled code is what could run. And so RNA, so DNA is the code. RNA is the stuff is the messenger RNA that ultimately gets expressed as signals and proteins. And so that's what's in RAM or in registers. And so we now have the ability to measure for the 20,000 genes that exist in the genome, a near continuous measure of how much each gene is expressed. So for 20,000 genes, I can tell you how much each gene is up or down. Following me so far? Okay. So what we did is we measured a bunch of kids who came to Children's Hospital for evaluation of inflammatory bowel disease. And these kids, and this is not my study, by the way, we just got the data. I'm not a GI doctor. And what they did is, which is routine, they look up your colon with a scope, and then they take a piece of tissue and they look at it under the microscope, and that defines whether you have inflammatory bowel disease or not. And so they did a bunch of kids, and labeled in red are the kids who, quote, ruled out or found not to have inflammatory bowel disease, and in the blue and green, those who did have inflammatory bowel disease. There's two types of them. One's called Crohn's disease, and one's called ulcerative colitis. The kid I'm telling a story about has ulcerative colitis. And as you can see, this is plotting each patient so each patient has 20,000 dimensions for the 20,000 genes, and this is simply a principal component projection in the first two principal components, which, as you see, actually capture quite a lot of signal. About 52% of the variants are captured in the first two principal components, and you see that, plausibly speaking, we've captured you could imagine a hyperplane. Right now it's a single line. We can imagine in the 20,000 dimension a hyperplane that does a pretty good job of separating the kids who ruled out from those who have the disease. And so before any of this had happened, we asked ourselves the following question. Could we actually find computationally drugs that would move these patients in that direction, from the disease to the health? This is where I have to start unpacking a lot of assumptions which may or may not be well founded. So one is, what does it actually mean to move? It means to move the RNA levels of the cells in this child. If gene N is low here and high here, we're going to push it down. If gene M is high here, is low here, and it's high there, we want to push it up. But basically, we're saying in this multi-dimensional framework, can we find drugs that do the linear algebra, essentially, to push that to there?
By the way, you have no particular interpretation of these dimensions other than this projection?
We've none. If you look down into several dimensions, I can actually tell you about which doctor it was, when it was in the study, and so on. So you can actually dissect. But the first two PCs seem to mostly capture. Seem to mostly capture measures of inflammation. Because actually, if you look at, I didn't want to bore you with these details, but if you look at the coefficients in the principal components, the 20,000 coefficient of the genes, and you see which ones are heavily loaded up front, have the bigger numbers, it's the genes that mediate inflammation. So the big distances here are caused by inflammation. Yes?
It's a naive medical question, but is the reason that we're just in genetic space here because we don't think environmental factors matter for these particular conditions, or is there like some weighting that is implicit in this too?
That's a doubly good question. A, we actually don't know the environmental factors, but we know that they exist. But we're not being that naive, because remember, we're not measuring here genetics. We're measuring here, so genetics is static. Your gene is static. The genes that are being expressed in your gut are very dynamic and responsive to the environment. So if there is, for the sake of argument, which I believe there is, microbial and dietary influences on the gut, they are going to affect the, so this, what they're doing, the gene expression is integrating the signal from the genetic program and the signal from the environment. That's what a gene expression program does.
To be sure, so the things that were, the diseases have been diagnosed were based only in symptoms. Yeah.
No, it was based on that pathological diagnosis. Look at the microscope. You have to count, I see this many white blood cells, blah, blah, blah.
Oh, okay, so that would be another dimension here, but it's.
Well, that's how we're
but
able to
it's
color them
It's
in.
the initial dimension. Okay,
Yeah, that's
it
the
doesn't
color.
give you an
It's not the
idea.
color, it's not the dimension, it's the color, it's the labels.
Right, okay, so it's based on.
Yes,
So in your answer to Maya's question, you're
Yes.
saying there's an epigenetic effect in the gut.
could well be.
Okay.
In fact, for other reasons, for sure.
Okay.
And yes,
So this blends in the DNA from the microbes as well, or is it like, how do you disentangle those?
so basically, it's a very complicated, but ultimately good metaphor for separating out by spinning around really, really fast, finding the things and chemically purifying it. So you just get the RNA, as opposed to human DNA or bug DNA. And so that's a
Alright,
short
so
answer.
this is purely human.
This is purely human. Great question. And in fact, since you asked a question and I only have a short time with you, I feel like I have to discharge myself of a lot of information. There's something called the Human Gut Project at UCSD, where if they do PCA on the microbes and not on the genes of the microbes, but they use the genes to phylogenetically type them, to say what species or phylum. And they do PCA, and they can actually show you when you have different bowel problems, your bowel flora goes in and out in PC space of different diagnostic areas. So someone who has something called C. difficile, which is a bad bacteria to have that causes mass diarrhea and kills people in hospitals all the time. You can see the gut flora change, and then when you treat it, it goes back. And in PC, multiple dimensional PC space, you see these multiple thousands of phyla go in and out together. But to return to our narrative, and again, I'm really thrilled that you're asking questions, and I'm just trying to charge forward just so I hope I can get to the punchline, which is not a punchline, it's a question for you. So, by the way, the one thing I want to point out, none of this analysis was done by anybody other than 3 PhDs in computational, with computational science, those 3 guys. I'll just say that no doctor I know could do this. And those are, one was a former grad student, and they were all postdocs of mine, but one was a former grad student, and the rest of them but had PhDs elsewhere. So, one last thing. So here's another hint. Why should we think that changing the transcriptome is going to change the health state? That's another assumption. Well, first of all, back to diabetes. Somehow, when I treat your blood sugar, even though it's not the cause, I'm bringing you back to health. And even though I'm not treating the underlying cause, there's also a longer bit of research, which I'll spare you, but it shows, for example, if you can take leukemia and make the cells, and you have a principal component in leukemia, analysis of leukemia and normal white blood cells, and you take the drugs that push the leukemia cells towards the mature white blood cells, the well-differentiated ones, you're picking out drugs that are A, pro-differentiation, B, actually clinically useful. So, in other words, messing with the code that's in RAM is actually changing the state of the executing program, aka the physiological state. Now, there's a lot of caveats around it, And so the good news is that there's a database created by one of my MD PhD interns, completely separately from me. He's at the Broad now and at the Dana-Farber, Todd Golub, called the connectivity map. And basically, in a factory-like way, they took thousands of drugs against a few cancer cell lines. And they said, we're going to measure 10,000 genes, expression, this is the dynamic measure, and we'll see how much each of those 10,000 genes get perturbed up and down by each of those drugs. So you have, for each of the 10,000 drugs, across the 10,000 genes, how much they're moving up and down these genes. So we asked ourselves, can we do the linear algebra? And crazy step number one, we're saying those cancer cell lines, these are gut cells. We say, nonetheless, because a cell is a cell is a cell, and the transcriptome, the stuff that's in RAM, about half the variables, half the RNA, are always the same across all different cells. Just to be a cell, you have to have a nucleus, endoplasmic reticulum, mitochondria, cell membrane. All that machinery is shared about 50% across cells. So we said, can we do the linear algebra to take the perturbation that we have from that database of the drugs onto the gene expression of those cancer cells, and then go to these individuals and say, if I applied that perturbation, would it push you towards healthfulness? And what was cool is it worked, in the sense that if you took a drug that's known to work for ulcerative colitis, it pushed everybody there. And if you took a bad drug like glucocorticoids, out here, when they're far away, actually the arrows would be pointing this direction.
This is in a test tube.
This is not a test tube.
This is a human, this is in people?
Oh, no, this is in a computer.
Oh, okay, right, yeah.
Right. So there was stuff done on cancer cells,
Right.
measurement of gene expression in humans, and these arrows are just linear algebra. Nothing has been done to animal, or no animal is harmed in the filming of this procedure. So this is purely computational, so keep that in mind. And so the arrows that you get computationally, going against the database that checked against thousands of perturbations against the cancer cell lines. If you look at glucocorticoids, here the arrows like this, here it looks pretty good, which, by the way, is consistent with our clinical experience, that if you're not very inflamed, they actually work well, and there is always pointing away. And although I don't have the slides show it, these students, postdocs, showed me something which I did not know clinically, but they showed me a drug that went like this. And what was that drug? It was zip medication, Accutane. And I did not know this, but when you look it up, Accutane actually causes, for some individuals, bad colitis. And so in silico, completely computationally, it looked interesting. But I'd never used it on a human being.
Couldn't you also take some particular linear combination of these and produce the optimal?
Yes. Yes. So we have an R frame. This is all R code. There's R code behind our studio framework behind this, and we have a Shiny app. This is all stuff out of a Shiny app. Dun, dun, dun. Yeah. So, by the way, I have to quickly say, I just taught a crazy course, computationally enabled medicine for doctors, for medical students, who I thought were going to be much more computer proficient than they were. When I was a medical student, people hissed when we talked about differential equations, but I thought this was 30 years later, it was going to be different. Turns out not. And so, luckily we had 6 TAs for this class. And
Only one student?
yeah. 6 TAs were barely enough for 24. And you see, they were smart enough, because they were Harvard medical students, they were smart enough to know this was a class to go to. So we had more students apply for this class than for any, but I told them they had to have no R and biostatistics. They basically lied to me. And so, but my point is, our only point is that the only thing that's made it doable, we had everything on Jupyter notebooks going against R in the cloud. So we were able to, if they screwed up their environment, as we said in a second, it was very valuable. In any case, so my friend comes to me and says, Zach, in 1 month my kid's going to have surgery. What can you do? So he had a month before gotten a repeat biopsy of his colon to see how he was doing. It was terrible. I said, take that and we're going to do gene expression, just like for these other kids, and see where he fits, and we'll do the linear algebra and see which is the top rated compound. And so it was storybook weird. The first postdoc that I showed there comes to me and says, Zach, this is the top rated thing. I don't know what it is. I said, I have no idea what that is either. In fact, it doesn't sound like a drug to me. And so I looked it up, and it's actually a supplement. And it's a supplement that's part of a group of compounds that make up indigo, which is a purple thing. And the Chinese, forgive me if my Chinese sounds like a swear word, Qingdai. I don't know, maybe it's the right way to say it, but I never know these days. And this Qingdai is used as a supplement in traditional Chinese medicine. And that was a top-rated compound that gave the biggest arrow for his kid. So then I looked up what I could find about Qingdai, and I found one article in a third-rate journal, unfortunately, and where it said that this Qingdai was useful in intractable ulcerative colitis. But I was well aware of the fact that there's a small number of widely used supplements. Now I know about Qingdai, but curcumin and so on, and they're tested for all these diseases. So just the fact that they did it does not necessarily mean that it was some special insight. So I called my friend and I said, this is the top rated compound. I said, I've never heard of it. It's a supplement. I did some research. It doesn't look too toxic. People take it. They seem to be okay. And
Give it to your kid.
and so I said, Zach, that's really weird because, I mean, he's a smart Harvard professor. He looked around. He said, I just talked to some people in Israel. And in addition to the standard Western medicine, they give Qingdai or indigo to all their patients. And so I said, well, that's great because now we really know it's probably not toxic. And furthermore,
all sorts of colitis.
colitis patients, yeah. And yes. And furthermore, the problem with supplements is the quality control is awful. I once did a study of DHEA, and you can see in the stores what you can get now, the composition goes from 100% to, using a mass spectrometer, 0 nanograms per sample. So you don't even know what you're giving. So the Israelis had done the quality control. They said, just have them FedEx over the indigo. And what happened, it's not a cure, but what happened, so this was December 2016. So within 2 weeks, this child went from pooping once an hour to pooping 4-5 times a day, started playing with dog, and within 2 weeks was back in school. And 14 months later, still fine. And everybody at Children's Hospital was ready, was geared to take his colon out. So here's my problem. The family's thankful. I look like a hero, but is this reproducible? Now, intuitively, I have to make it clear. This really looked bad. The colon on biopsy looked bad. The kid was doing poorly. All our expensive drugs did not work. But the challenge to you is, what trial could we possibly do to convince ourselves that this works? Let me phrase it a bit more specifically. What is the minimum possible trial? What is the MVP trial, minimal viable product trial, that would prove to, go ahead.
Why not, after the initial biopsy, I mean, couldn't you have run the original set of drugs that were tried first on the very first biopsy?
So,
Or is like I'm sorry, I'm not a doctor. To play one on TV on occasion.
yeah,
The
and very well, I should say.
But like, in some senses, you expose this patient to a whole series
Yeah,
of medicines, like just trying things out.
Yeah.
Why not the very first biopsy? You get enough material, and then crank it through your, expression procedure, and then you would have ruled out having to give them all of these, crazy anti-inflammatories early on.
excellent question. Let me give you a couple of answers. First of all, and these answers, which will sound obvious after the fact, were not actually obvious to me when I was in the heat of battle here. One is, what distinguishes why can I even do this crazy thing, as opposed to the beginning? Because at the beginning, we actually have treatments that work for most people. And in fact, it worked, a very mild anti-inflammatory, worked for this kid for 10 years. So, and much less toxic than all the other things that we tried afterwards. So he was in a different physiological state. So if you think of these systems as systems that are stateful, his state was very different. Second is, it's unethical to drop things on people that have possible toxic effects, especially where you don't know, unless the utility functions start getting very, very significant. And here they were getting very, very significant. Losing a colon is a big deal. And so this is where it comes to rare disease, because the point is, I told you about Undiagnosed Disease Network, and we've actually, through this network, we've literally made hundreds of new diagnoses, new entities. But the point I want to make is, a lot of our common diseases, diabetes, inflammatory bowel disease, buried in there are plenty of undiagnosed diseases. We just have to give them a label, but they're actually not the real diagnosis. How do we know? Because they don't respond. So back to my precision medicine metaphor, in one axis, signs and symptoms, they're fitting perfectly the definition. But in multiple other axes, they're not. And how do I know that's true from our patient? A, I just showed you, didn't respond to every therapy that we had. B, after the fact, we did some genome sequencing, and I found some mutations in this kid that affected the white blood cells that other people don't have. And so I'm just telling you, part of medicine is that there's much more of us who are undiagnosed than we think. We have these labels and we sit with them because what do we know? We're just civilians or medical doctors who take our pablum for granted. But in fact, big chunks of us don't have common diseases. We have these rare forms. Guy in purple.
Well, sorry.
Yeah, one. Dr. T.
I guess my point was, why not biopsy first,
We did.
right? Get that, expose that biopsy to computationally
Yes.
to the whole range of the drugs that you tried first.
Yes.
And the whole range of other things that you just threw at it, including the indigo stuff.
Yeah.
And just start there.
So I can tell you that we did do that, and it worked great for the drugs that he started with. We had nothing to add.
Right. But then he shifted
He
states.
shifted state with puberty.
Got it. So he hits puberty, gets biopsied again,
Yeah.
and then run through the same
Yeah.
gamut.
Did your statistics or
Yeah.
data indicate that he changed states when he went through puberty?
Yes.
Okay.
Yeah. I mean, but the problem is confounded because just being
Yeah,
older.
because of a million things, right?
Well, like a million things, like being older.
Right.
But we know that that having growth hormone and testosterone, there's receptors for those hormones on the hundreds of genes. So just knowing nothing still tells you it's going to be a different
The
state.
purple guy's Max.
Yeah.
Max.
Max.
How
Max, thank you.
When
At first I wrote like a maximum function. Yes.
So how distinctive was the indigo thing? Was it like very clearly the best thing, or were there lots of things that were all about as good?
So it was actually, I'd say, I didn't do this in a Bayesian framework, but if it were a Bayesian factor, I bet the Bayesian factor would be like 2. It was a big jump, and then there was a whole bunch of things stacked.
Right.
And then yeah, so it was actually pretty different, and it was not what I was expecting because it was not a drug.
Yeah.
Yes.
So,
Yes.
so I just looked up Qingdai on the
Oh, now I'm really.
Do you pronounce it right? It's called Qingdai. It
Good.
has a
Thank
lot-
you.
It has a lot of functions, like like drawing eyebrows,
Okay.
and also there's like snake bite, or like something with children, or like vomit blood, and there's lots of things
Okay.
like that.
He has nice eyebrows, and those haven't changed. So good to know. You know what? I have done it. I appreciate it. Seriously. You're saying?
Yeah, so if you said that, for example, in a trial that was given, Qingdai was given,
Yeah.
and it was positive
Yes.
result, right? If you get a database of worldwide of treatments, given the symptoms at that point or the state, would you have come up? Do you think we would have come up with the same response? I mean,
That is
probably-
that is part of the question. That is part of the question because, again, the order, it may be order dependent. If this place is stateful, the order in which you give these things might matter.
But the answer to this is quite straightforward. It's
Okay.
not
That's-
Well,
So that's why they can't big box.
Statistically.
Go ahead, go ahead.
Statistically, okay. So the answer is not to give indigo to everybody who
Yeah,
has this disease.
right.
The answer is not to give everybody to indigo, or my answer is not to give indigo to everybody with the disease who seems to have the same characteristics. It's to follow your procedure. You have an algorithm. You're
biopsy
on
first.
Is that what you said?
Yeah, you learned this by taking a biopsy and then doing
Okay,
exposure.
but okay, guys, but by induction, what is the inductive principle, which will, because I'm giving you an entertaining story, and so it's somewhat believable, but now put on your FDA hat.
No, you have an estimator.
As an estimator, and how do I get the estimator closer to where you find it believable?
Take a sample of people.
What kind of sample, on what
Well,
statistic?
people who are desperate enough to go to a computer science PhD for a diagnosis.
Turns out it's actually not that hard to find.
That's right, that's right. So take them,
Yeah.
since you know something about ulcerative
Good,
colitis,
good.
do it in that
Yeah,
area.
yeah,
You have all these sites, they're doing different
Yeah.
things, so, which obviously indicates they have no idea what's going on.
Right.
They'd be willing to let you do it.
yeah.
So you take all those, you follow exactly your procedure, you come up with the best linear combination, you recommend a drug.
Right.
And then you measure the change in how they feel.
Okay, but
Or whatever.
okay, so let me phrase it in a narrow applied statistic. What survival function am I comparing this against? You know, what am I comparing this against? Now, on the one hand, I think that most of these patients fail. I think it's obvious this is working because it wasn't working before.
Well, you have
But the skeptic will say, Zach, there's some probability that just by chance this kid would get better. That's
You can only do this for a subset of all the people who have undiagnosed ulcerative
Right.
colitis, whatever it is. So you randomly select the ones into your sample, you randomly select the ones not into your sample as the control group, and see whether one group is better than the other. It's a randomized clinical trial.
but what is the figure of error that we're trying to maximize?
You have to measure the outcome.
Yeah,
Number of
what is the
times-
outcome? Is it, you
Number
know,
of times in a
Number of
group a
poops
day
per day.
is what you
Poops
told
per
us.
PPD.
But clearly, if you're gonna do something like this, you come up with as many outcomes as possible, right?
Yeah.
Because it's the same, for the
So,
same price.
so we've already, I think, reproduced a lot of my questions, because the standard of care is highly variable, the specific interventions are variable, the ordering of interventions is variable. Individual cases will have shared and unique pathophysiological states. They'll have some things in common, some things not. Individual will have shared and unique predispositions, so they'll have both unique exposures and unique genetic characteristics. So in some ways, the conditioning matrix is very sparse, which is also
Always
problematic.
the problem.
What?
Which is always the problem in this area.
Yeah, yeah. And so, and specific effective agents will therefore vary from case to case. And not only that, but this is a moving target. This changes with age. This is actually work I did with Finale Doshi at the School of Engineering and Applied Sciences. She was doing a nice job of showing that an incidence of inflammatory bowel disease increased in autism. But the black curve is in these three large databases. It just shows the age, the cumulative distribution of cases of inflammatory bowel disease. So let's discuss how to show this is a reasonable approach. So I think one way would be to say, at how many surgical interventions could I avoid? Might be one thing to do, because that's what we find in medicine is that intermediate outcomes are always gained. In fact, they're gained by pharmaceutical companies a lot. So one of my statistician colleagues, who you may or may not know, at the School of Public Health, L.J. Way, always talk about the fact that for him there's only one outcome measure, and that's death, because that's hard to gauge. Everything else, you can move things around. Surgery is kind of like that, in the sense that you either had surgery or you didn't. It's hard to argue. And so let me quickly just share with you something that we did with one of the three postdocs I showed you. And basically, we looked with this amazing database through an insurer that has, in fact, it's Aetna. And although the book of business may be 20 million people, we have all their business over many, many years, so that we have many more patients than they have subscribers at any given point. So we have 60 million patients. And so we went through all the ulcerative colitis patients and went all the ones who had inflammatory bowel disease who ultimately had surgery, and we basically clustered their temporal courses over time. We identified 8 clusters, and I can go on about how we decided to find 8 clusters as opposed to 4 clusters or 10 clusters, but we found 8 clusters of these patients, and we characterized them in a variety of ways.
Sorry, are these clusters from the procedure that you had, or like what?
These are, sorry, I was unclear. So now, reset. We're talking now a bunch about patients with inflammatory bowel disease in a large insurer database. Same inflammatory bowel disease, ulcerative colitis. And we're saying, can we group them so we can start creating predictors around them? Why do we want predictors? Because if we have predictors, we can say, if you're in this state, I expect you to be there in the future. And that would be a good way to compare our intervention against there. That was my answer to your question, to the question I posed to you. And but wait, just so you're not disappointed, I haven't answered my own question, but I'll show you how far we've gone.
That one requires or depends upon the accuracy of your prediction.
Correct. And you'll see a bit. So we've taken this patient population and basically put everything into the machine, and we're able to create these 8 clusters that are quite distinct, and then we use them predictively. So we in the sense that we label each patient as being as a trajectory through these states, and then we say, can we predict when they're going to have surgery? And we ask ourselves, how many months of data do we have to be able to predict X months in advance? And this is the accuracies that we got. So on the bottom is months before surgery, and the y-axis is how many months we'd observed the patient at the time we made the prediction. So this is how long have we known the patient, so how much experience do we have with the patient, and this is how much before the event, the surgery event. And you can see it doesn't do too badly. And again, we have not used this, but I'm just telling you that you can start using this data predictively to say, this is what you can expect. And guess what? These are predictions that no surgeon can actually do. They don't know this. In fact, I showed it to some surgeons. They want to use it, obviously. And so, I think that in this world of a sparse matrix, precision medicine, I showed you multiple essentially matrices stacked on top of each other to create this multidimensional matrix. It's always going to be sparse. And so I think thinking of how do we do trials across these sparse matrices is a huge area of applied statistics that some of which the methodology is done, but the applications are fiddly, and some of which the statistics are not well done. And we have to get them well done enough so that we can, and popularized, so that we can actually run it by places like the FDA. I think that's where the future is. I don't know if you've heard one of my colleagues, Matt Might, speak. So Matt Might was a computer science professor, and half the story appeared in the New Yorker. He was a computer science professor, tenure track, University of Utah. Child is born, first child is born, floppy, doesn't develop that well, gets worse, starts having seizures multiple times a day. Utah has a great genetics department. They look at him, make several diagnoses. All of them end up being falsified. And then he, using his network, reaches out to people doing early exome sequencing in 2008. A guy called Goldstein, who was then in Duke, now in Columbia, and they sequenced his exome, and they found him and proved that it was a mutation in gene never previously associated with disease in humankind, called NGLY1. And that's why the New Yorker article is called One of a Kind. He also, this computer science professor, had a blog. So he had one patient, so he needed to create a cohort. So he blogged and shows Liam Neeson and Taken with a gun pointed right at the camera. He says, I was looking for the killer of my child, and it was me. And you see him and his wife contributed 2 different variants that together make this kid homozygous. And he also rented Google AdWords. This child was seizing, floppy, and didn't have no tears, had no tears, a lacrima. And although it's for porn words, you can spend several dollars in AdWords, but for alacrity, it's just a cent. And so just using Google AdWords, he was able to find 50 patients within a couple years. And so I tell my medical students, how many doctors do you know who have A, diagnosed disease, and B, found the population? And then ultimately, he did a bunch of studying up, and while looking for very expensive gene therapy, he actually figured out with some enzymologist that this child might be missing a simple sugar monosaccharide in the brain. And he looked it up on Amazon, and and I saw him tell the story right in front of the FDA commissioner. And so that stuff costs cents per pound. And so he got it, made a big shake of it, drank it, did not die. The next day, he gave it to his kid, and the kid for the first time ever had tears, and stopped, and the seizures went way down. And so his wife has worked on right to try legislation, and he works with me now on this undiagnosed disease network, and he did because of a conference I invited him to, he ended up working for the White House. In fact, he was
right?
What?
He's at Alabama now, right?
Yes,
He
now
did
that-
data symposium.
Right.
Data science symposium
Exactly.
a couple months ago.
Right.
And
So Matt was the last. He told me, he gave us nice tours of the White House because I helped him get there, and he basically shut the door on the Office of Science and Technology, OSTP, because OSTP was big out there in the White House, and in the Obama administration, he was there, and then it just basically did not get renewed, and he was the last person out the door. That he just left about 2 months ago. But my point is, he now went to strong right turn from computer science, programming languages, crypto, to being the head of a precision medicine institute at the University of Alabama. And these kind of issues about how methodologically how to do these rare trials is up front and center.
How important, given your understanding of the mechanisms of some of these drugs, is understanding the potential for their interaction, right? So you just showed me that you have these
Yeah.
8 different clusters.
Yeah.
Does that ever come up? I mean, 'cause that increases the sort of sparsity
Sure.
even more,
So
even
I think,
with just two-way
I
interactions.
think you start from the perspective that I have to remind my former PhD advisor, because he always forgets how much medicine sucks. And so, given the fact that you really internalize that medicine sucks, that it doesn't know what it's doing, first order theories should be first tried before you do second order effects. And so, just trying to unsubtle things, the one versus the combos, it is probably better justified than the way most medications are actually chosen in these patients. You just don't understand how bad it is. I mean, it's not that doctors are mean, it's just that we know very little, and the way we're taught medicine, we don't really acknowledge it to ourselves, let alone to our patients. But here's the thing, we have a department at Harvard, we have lots of great data, with lots of great questions. I think I'm on your SAB of Institute. There's lots, we're working together on the data science initiative. So, and today I'm going to be marrying one of our the Data Science Institute has initiative has postdocs, and we have 7 postdocs, one of which is working with me, and with I forgot his name, statistics. And Chinese won't help. When I say Chinese, it won't help you. He's now in Singapore. What? Who?
Sam.
Yeah, Sam. Who said Sam? Thank you. Sam he's in Singapore, and so this guy is marrying his sweetheart from Taiwan. He did MD, PhD. PhD was at Stanford, MD in Taiwan, and he's doing this kind of integration, doing deep learning. Sadly, deep learning is for me a sore point because it works way better than it should. And I was always about probabilistic frameworks, and it's killing us. But anyway, he's using deep learning to really outperform pathologists. He's looking at pathology images. Anyway, so I'll be 2 hours. I'll be wedding the 2 of them. Get something called a solemnizer certificate from the state of Massachusetts. It gives you authority to marry people for 24 hours. So listen, thank you very much for your attention.